Okay, hello everyone. Good morning, good afternoon. Welcome to the third Covance Arcinova presentation, Recent Insights for Optimizing Your ADME Human AME Study. Very pleased that you could join us. My name is John Headley, and I'll be the moderator for today's call. Before I introduce our presenters for today, just some sort of housekeeping, general guidance for the call. Our presentations will last approximately 50-55 minutes. We're very happy to take questions, and we'll take these questions at the end of the presentation. So what I would ask for colleagues is, if you have a question during the presentations, please use the chat function at the bottom right-hand side of the screen and send your question to me directly, and I'll read out the questions at the end of the presentations. So we're scheduled for the call to last for an hour, but we're very happy to carry on for 15-20 minutes after the top of the hour, just to make sure that all questions are answered. So please feel free to send me those questions during the presentations. And then later on today, we'll be sending out the presentation to all attendees, so you'll have a copy of presentations from today. So if I can move on to introduce you to the presenters for today. Firstly, Catherine Laing. So Kath is the Associate Scientific Director at Covance, worked at Covance for over 18 years, based in our clinical pharmacology group in Leeds in the UK. A number of roles, including PK, project management, and medical and scientific affairs, has been a scientific reviewer for a wide range of clinical pharmacology studies, and has got significant experience in optimizing phase 1 study designs. We also have John Kendrick, who's the Director of Science and Commercial Strategy within the Department of Metabolism and Environmental Sciences at Covance. So John's responsible for the oversights of innovation and scientific guidance to clients on all aspects of DMPK in drug development. John's been with Covance for about 14 years, in a variety of leadership roles, with global oversight of the radio-labeled ADME human AME study portfolio. Prior to Covance, John was at Eleanor Row as a laboratory and study manager, and oversaw development of microdose and microtracer studies for a wide range of molecules. And we also have Eleanor Row. Ellie works for our partner organisation Arcinova, where she's head of client services. Again, with over 15 years of experience in pharmaceutical and contract research environment, working with multinational companies such as Sanofi Aventis, and also Covance, where Ellie was a senior radiochemist for over 10 years. Ellie also has significant project management experience and a proven track record of delivering projects globally. So these are our presenters. We'll work through presentation for the next 50 minutes or so, and as I say, welcome any questions, and please send me those through the chat function. I'll be very happy to ask them. So John, I think I'll hand over to you.
Thanks, John, and good afternoon, everyone. So themes running through today's WebEx will encompass a brief regulatory overview of what changes have driven the importance of conducting human AME studies during drug development over the last 20 years or so. And we'll provide a set of indicative timelines that help with the planning and management of expectations with key milestones along the human AME continuum. We'll also then introduce some of the non-clinical prerequisites that govern the setting of a safe radioactivity dose that can be administered, before moving through challenges and considerations on provision and use of the radio-labeled material. We'll change gears a little then and review the notable pharmacy contributions and the clinical conduct elements, before rounding off with the analytical endpoints of a human AME study, and that's namely the radioanalysis and the metabolite identification. And we'll close with an example of how a coordinated collaboration is integral to the successful human AME outcome. Now, at the end of the WebEx, we're going to keep lines open for Q&A as John described, and hopefully by then we would have already touched upon such questions as, what are the scientific and the regulatory requirements of a human AME study? What are the likely pitfalls and the largest challenges that we encounter along the way? How long does it take to plan and conduct a study? And what information does using a radioactive dose provide over a conventional non-labeled clinical study? Now, the answer to that last question is illustrated in part here. These data are taken from blood and plasma analysis for radioactivity by liquid scintillation counting and for unchanged parent compound measured by standard bioanalytical LCMS. By comparison of the Cmax values for total plasma radioactivity against those of the parent concentrations, it's evident that there's a high first-pass metabolism involved for this molecule. In addition, the 25-fold higher half-life for total radioactivity compared to parent highlights the presence of long-lived plasma metabolites. Consequently, systemic exposure of radioactivity is around 25-fold higher than parent alone and highlights that these long-lived plasma metabolites are circulating at high levels and contribute significantly to the metabolite profile of this compound in humans. And this, of course, dictates further regulatory consideration. So what are the regulatory kind of guidelines and current regulatory position for the design and conduct of human radiolabeled studies? Now, all global agencies involved in drug development have moved towards a greater understanding of the metabolism of a drug before large-scale patient studies, and this continues to be driven earlier in development than ever before. This has focused considerably on the scientific and safety of unique or disproportionate metabolites, and of course the rates and routes of elimination of a drug and its metabolites during those investigations. But extensive understanding of the metabolism of a drug and its potential for drug-drug interactions is also now required prior to any first-in-human study, particularly within the US. You can see that the frequency of the publication of guidance updates has been increasing over recent years, and bit by bit the global regulatory agencies are becoming more closely aligned with each iteration of their own literature. Key guidances are the ICHM 3R2 guidance, which was first published in 2008, where a need to understand as completely as possible the metabolism of a drug is highlighted, with PK data relevant to potential drug interactions required before phase 3 being notable there. It was also highlighted that there was a need to compare human and animal metabolites and determine if additional testing was warranted in the document for the first time. The EMA drug-drug interaction guidance from 2012 described total radioactivity, or the total recovery of radioactivity in urine and feces should exceed 90% of the dose administered in a human mass balance study. And moreover, greater than 80% of the recovered radioactivity should then be identified, i.e. you're characterizing the metabolites. And of course, the misguidance in 2008, 2016, and recently updated in 2020, of course, states that characterization of human metabolites is warranted when metabolites are observed at exposures greater than 10% of the total drug-related material. And furthermore, initial metabolism investigations should be conducted before initiation of the clinical trials. So what's needed to organize and complete a successful, human AME study? And this could be answered by providing an overview and of course an appreciation of the interconnectivity of the key components that span the planning of a human AME study, from the radiosynthesis to the non-clinical studies, from the clinical conduct, the analytical involvement, and of course the openness to be innovative in the generation and evaluation of the data obtained that address both the regulatory and safety questions. And we'll go more into detail as we move through the session today. Consideration has to be given to the time and sequence that each of the key components involved in a human AME study takes when planning is initiated. This includes consideration of geographical locations of not only the clinical phase, but also the API and the IMP manufacture and testing, as well as the downstream radioanalysis and analytical endpoints. And of course, who should be responsible for the coordination of these activities is also key. As already alluded to, this is often a multidisciplinary, multi-site collaboration, and coordinating these efforts is crucial. The non-clinical radiosynthesis is the first activity that initiates the cascade in laboratory-based events. As a preferred partner of Covance, Arcinova would often be engaged in tandem with us in early discussions with prospective clients on the requirements of the non-clinical carbon-14 synthesis. Also in the planning and delivery of those materials to Covance. And then finally, the timing of the preclinical animal studies is carefully coordinated. The team at Covance in Harrogate in the UK, Madison in the US, or in Huntingdon in the UK would be the location for the preclinical laboratory conduct. And the Covance Clinical Research Unit in Leeds in the UK or in Madison in the US for healthy volunteer studies, or alternatively, a third-party clinic such as that at the Royal Liverpool Hospital for oncology patient studies would be the site for clinical conduct. And then finally, radioanalysis and metabolite profiling and identification would once more fall under the remit of the Covance DMPK labs in either Harrogate, Madison, or Huntingdon. So I'll now hand over to Ellie to describe the radiosynthesis requirements in a little bit more detail.
Thanks, John. Good afternoon, everybody. I'm really pleased to join you today and just to talk through some of the key considerations in planning your ADME and AME studies from a radiosynthesis perspective. I'll go into each of the sections highlighted on the slide in more detail and really hopefully provide you with an insight into aspects around labelling strategies, timings, stability of radiolabeled compounds, the uses of different batches of material, quality standards associated with them, and also any benefits that can be gained for planning for these studies at the same time. So the first thing we really need to consider is the labelling strategy, and determining the most appropriate place to incorporate the carbon-14 radio label is key as it impacts the timing and the cost of the synthesis. Ideally, we would incorporate the radio label into a metabolically stable position, which would allow us to follow the majority of the metabolites circulating in plasma, urine, and other matrices. Occasionally, we do see compounds that have a tendency to cleave down a central bond, resulting in two different and distinct metabolic pathways. And in these cases, multiple labels may be required to follow both the left and right-hand side of the molecule to ensure that all the metabolites are identified. And it's this labelling strategy that really drives the timelines, as the duration is determined by the availability of the radiolabeled starting material, the number of synthetic stages, complexity of the chemistry, and ease of isolation. As a radiochemist, the number of commercially available starting materials is quite limited, and unlike medchem or process chemistry routes, we're really restricted to a few very simple building blocks, with the majority of syntheses starting from radiolabeled carbon dioxide, potassium cyanide, or U-labeled aromatics such as aniline. This often results in a synthesis which is longer than that of the unlabeled counterpart. So when we're designing the synthetic route, we find it's often a trade-off between incorporating the radiolabel towards the end of the synthesis to reduce the costs, and having the radiolabel in the core or more metabolically stable position of the compound to give you better non-clinical and clinical data. The specific activity is dependent on the study type, and non-clinical studies are usually conducted at high specific activity, and depending on the molecular weight of the compound, materials are usually supplied between 3 to 5 megabecquerels per milligram. However, your clinical studies need to be performed within the limit of the safe radioactive dose, as determined by the dosimetry data. But again, there's a balance of needs here as well between the study objectives and the synthesis or stability of the radiolabeled material. Where a higher radioactive dose may support better clinical data, it can also contribute to higher synthetic costs and possibly lower stability and a shorter shelf life. And as many of you will be aware, carbon-14 compounds often exhibit different impurity and stability profiles to their unlabeled counterparts. So it's really important that we perform a stability assessment of the material at the correct specific activity to ensure the clinical shelf life. Typically, we'll run stability studies for around 3 months, which is usually sufficient to cover the release testing, shipment of the radiolabeled material, the drug product manufacture, and dosing. although these stability studies can be extended if the study is to be run in patients rather than healthy volunteers, and we may go out to around 12 months to cover recruitment rates. We also like to think about the route of administration early on, as it really does affect the release testing requirements of the API and the amount of material required for analysis. Depending on the specific activity, this can add considerable cost to a synthesis, especially if it's a long, low-yielding synthesis and microbiological testing is also required. Also, human AME studies are often conducted when the compound is in a later phase of development, where the formulation may have moved on. So a simple fit-for-purpose formulation, such as a powder in bottle, which can be constituted as an oral suspension or solution, or a powder in capsule formulation can be developed in parallel, including consideration of where the drug product will be manufactured. And this can be either at Arcinova or prepared extemporaneously at the Leeds clinical unit just prior to dosing, both of which have their merits, and it's really up to our client's discretion as to their preferred site of manufacture. And finally, from a regulatory perspective, all of this information relating to the synthesis, the analytical methods we use, the stability data, so principally your initial and 1 month time point, needs to be compiled into the CMC dossier sections, and this is for inclusion in the IMPD submission. So, as I've alluded to, there are lots of considerations, and it typically takes around 10 to 11 months from initiating the radiosynthesis through to your clinical start date. The work is usually conducted in 2 phases, one for the GMP, non-GMP, and one for GMP. Initially, we'll perform a trial synthesis, which is conducted at low specific activity. So here, the unlabeled materials are spiked with a small amount of radioactivity, and we use this trial to confirm that the chemistry works well in our hands and to satisfy any environmental regulations. It can also be used to determine the most appropriate hold point for the GMP starting material. Following this trial, the chemistry is repeated with high specific activity material, and at this stage we'll often store a portion of a late stage intermediate for the GMP synthesis, which is beneficial from both the cost and timing perspective, as it removes the need for a second complete synthesis and takes the GMP stages off the critical path. The remaining material is taken through the final stage to supply material for non-clinical and QWBA studies. The data from your QWBA study is really important for the next stage of the radiosynthesis, as it defines the radiochemical dose and therefore the specific activity of the material we need to produce for the technical batch and GMP synthesis. Once we have this information, a technical batch is prepared and a stability study conducted to define the shelf life of the drug product. However, if your radiochemical dose is very low, the technical batch will often be prepared at a minimum of 1 millicurie per milligram. This will be considered as representative for your study, but will just enable us to do a more accurate determination of the radiochemical purity. Where the drug product is a powder in bottle, which is due to be constituted at the clinical unit, the API can be stored in the same primary containment, thus supporting the shelf life for both the drug substance and your IMP, with only an in-use stability, often around up to 72 hours, being required for the constituted product. The final stages of the GMP synthesis take around 6 weeks, and we'll do a repurification of the labelled starting material, the final stage of chemistry, and an isotopic dilution with unlabelled GMP material for supply to the clinic. Once this and the material for the IMPD has been submitted, we usually plan for around 20 to 30 calendar days for the IMPD review by the MHRA. When it comes to clinical studies, the most commonly asked questions we get are, Can I use non-GMP material for my human AME study? And how do you determine where to start the GMP synthesis? Unfortunately, this isn't straightforward and is dependent on 3 factors: your radiochemical dose, the isotopic dilution, and the type of formulation to be administered. So as you can see from the diagram on the slides, the approach we take is often determined on a case-by-case basis. So if we take a standard radiochemical dose, which is usually around 100 microcuries, where the material forms more than 10% of the dose, so a very low isotopic dilution, regardless of whether it's for oral or IV administration, we would go back to a late-stage intermediate and perform the last carbon-carbon bond-forming step to form the API. We'd also do this followed by a purification and the isotopic dilution with GMP-grade unlabeled material, and these 3 stages will be performed in the GMP laboratory under GMP conditions. However, if your radioactive dose is less than 10% and the material is for intravenous administration, the same approach would be applied. If the material is to be administered orally or as a microdose, and there's a very high dilution of unlabeled API, then materials from the non-GMP synthesis can be repurified and diluted with unlabeled GMP material, thus reducing your number of GMP stages. There are some caveats to this, however, and any materials that go into the GMP synthesis must be traceable and come with a BSE TSE statement and a certificate of analysis. The GMP materials supplied is prepared in a dedicated laboratory using equipment managed under our QMS system. Raw materials are always independently released by quality control, with QA having oversight of the process and any deviations or changes. Unlike the non-GMP API, material is released using validated methods against approved specification and is supplied with a certificate of analysis and a certificate of release issued by QA. All materials are shipped to the clinical unit under a controlled chain of custody with temperature logger and tamper evidence seal. So the Covance clinical unit will receive around 5 millicuries of GMP grade material at the correct specific activity, which is sufficient to perform any mock preparations and the final clinical batch. Alternatively, should a client wish a fully QP released IMP, this can also be supplied directly to the clinical unit. Hopefully, this has given you a good oversight from the radiosynthesis point of view and has highlighted some of the key challenges faced and the advantages of planning ahead. I'll now hand back over to John for further insights into the ADME and QWBA studies.
Thanks, Ellie. So the non-clinical animal studies are aligned with the provision of the radiolabeled API, as Ellie described, and these typically take anywhere between 2 and 4 months to complete. To complete a simple rat study with a single dose route to longer time frames for more complex studies with multiple dose levels or multiple routes. Or if bespoke investigation phases are included, then that can obviously increase the timeline. So if we break it down into its kind of constitutive elements, the only necessary preclinical ADME studies required to support the radiolabeled human AME study are a rodent tissue distribution study, typically a QWBA or quantitative whole body autoradiography, and a rodent excretion balance study. And 90% of these ADME studies are in rats using a similar strain to those utilized in the toxicology studies, with mouse being the alternative rodent usually selected. Now, large animal studies are often conducted in parallel or sequentially to the rodent ADME study to obtain further species-specific PK, metabolite, and excretion data, maximizing the use of the radiolabeled material available and prepared for the non-clinical rat ADME study. But these are not actually required for the critical dosimetry calculation and are therefore not on the critical path. The rat ADME study does allow the data from that tissue distribution and excretion phases to be used to estimate that safe dose of radioactivity that can be administered, as Ellie alluded to. And this calculation is reviewed by Covance in the first instance And then a recommendation is submitted to RSAC, which is the Administration of Radioactive Substances Safety Committee in the UK, for approval in the human AME trial. So if we break the dosimetry and the tissue distribution phase down a little bit more, what do we have? Well, some important aspects to consider when designing the preclinical ADME studies are that the same radioactive isotopes should be used in the animal studies as the intended human trial. And this is because differing isotopes have different properties and therefore have different risks to tissue and body burden in terms of that radioactivity. The data generated from the animal studies should be in the same gender as that intended for inclusion in the human study. And typically males are used in radiolabeled studies, but if women are also to be included, or the therapeutic area is relevant to women, then females or female animals should be included too. The route of administration intended for the clinical trial should also be mirrored in the animal studies. So an oral dose in humans will need an oral dose in animals, assessing tissue distribution and the rates and routes of elimination. Additional data, such as absolute bioavailability after IV administration in animals, is useful to obtain at the time of the ADME studies, but it's not critical to the conduct of the human AME study, unless, of course, an IV route is also intended. As you'd expect, the time course of a tissue distribution study, as with any biodistribution investigation, should cover an appropriate period so that the absorption, the exposure, and the elimination characteristics can be evaluated thoroughly to enable an accurate dosimetry calculation to be performed. And with the tissue distribution study, all critical tissues covered under that dosimetry calculation guidelines prepared by the WHO and the ICRP need to be assessed, where the radioactivity is quantified and quantified over an appropriate period. And this is usually minimally a week, 168 hours. Finally, binding to pigmented tissues of the eye and skin, which are also part of that critical tissue list when conducting the dosimetry calculation, should also be included in the study. Therefore, a QWBA study often includes the toxicological strain of rats, such as the Han Wistar or Sprague Dawley, but also a pigmented strain of animals too, to provide bridging data into that pigmented tissue. And these would include something like the Long-Evans or the Lister-Hooded strain of rat. So QWBA and excretion data are taken, and using validated weighted formulae, a safe radioactive dose is scaled from that animal study or those animal studies to the humans for use in the consideration of the human AME trial. So when you have an indication of the radioactive dose level that can be administered to humans safely, this is not the end of that particular story. After all, it is critical that the dose administered enables the objectives of the human AME study to be met with the lowest possible dose. So for those that have been involved in human AME studies previously, a typical radioactive dose is 100 microcuries or 3.7 megabecquerels, as Ellie described. However, dosimetry calculations often return a value of between 60 and 80 microcuries that fall within both the guidelines of the WHO, the category 2, and the ICRP, category 2A, up to 1 millisievert. And to comply with both regulations, that is the upper limit. In the UK, we've often justified, though, increasing the radioactive dose to provide an effective dose of greater than 1 millisievert. And this is based upon the need to increase above that 100 microcurie benchmark to ensure we can meet the scientific endpoints of the study. And these 2A and 2B categories represent a minor to intermediate risk in terms of the radioactivity exposure and the risk of cancer from that radiation, which are considered acceptable in terms of the conduct of a clinical human AME study. And Kath will touch on this in a little bit more detail shortly. So I'll hand over to Kath for an introduction into the regulatory considerations in more detail involved in that clinical trial.
Thanks, John. So in this section, as John mentioned, I'm going to touch upon the regulatory submissions themselves, the documentation that is required to be submitted to the regulation to each different committee. So the process of itself takes around 6 weeks. So we'll look in a little bit more detail on the next slide about the different documentation and different submissions that need to be made. So, as I mentioned, there's a number of different submissions that are required prior to being able to initiate a human AME study in the UK. All of the documentation that are required can be prepared in parallel to the radiosynthesis, and so you can be ready to make the submissions as soon as all the data is available. So, as with all clinical trials, a CTA submission needs to be made to the MHRA. And the main difference really between the submission for a human AME study compared to other clinical pharmacology studies within the programme is the IMPD. So the IMPD needs to contain all of the necessary supporting data for the 14C API, in addition to the 14C drug product. So this review process can take 3 to 6 weeks by the MHRA, and that really depends on whether or not they have comments back on the submission. And so if they do have comments, then the review process will take 6 weeks. In parallel to the CTA submission, the ethics submission can take place. So this is made via the Integrated Research Application System, the IRAS system, in the same way as it is for all clinical pharmacology studies. And the documentation is submitted to a committee who are used to reviewing phase 1 healthy volunteer studies. So in parallel to these sort of standard submissions, for a human AME study, we also have the RSAC submission. So as John mentioned, this is the Administration of Radio-Labelled Substance Advisory Committee. So their role really is to review the radiation exposure that the trial subjects are to receive and determine whether it's within acceptable limits. So the application is made through the IRAS system in the same way as the ethics submission, and therefore the RSAC committee receive all of the same documentation that the ethics committee will receive. So in addition, there is an RSAC form that this committee review, which contains an overview of and the purpose of the research, as well as details of the radioactive material which has been extracted from the dosimetry report. There's also a risk assessment within this form, which is reviewed and approved by a medical physics expert who is familiar with the site, as well as a clinical radiation expert who is the license holder for the radioactive administration at the site. So the RSAC review itself can take slightly longer than the ethics and MHRA review. It can take up to 8 weeks, although the approval is generally received much quicker if there are no queries. But just to note that the RSAC approval is only required for dosing, so not for screening, and so therefore it's not usually on the critical path for the study. So you're able to start the recruitment of the subjects in parallel with the CTA and ethics reviews, so that as soon as you have those approvals in, then screening can start while still waiting for the RSAC approval if that hasn't been received. So now I'm going to move on and talk a little bit more about the clinical phase itself. So looking at the study design, the dosing, the sample collection, and also the radioanalysis. So the design of a human AME study is fairly standard in order to meet the standard list of objectives that these studies have. However, there are some additional considerations that needed to be taken into account on a case-by-case basis. So the typical objectives that we have for these studies are to assess the absorption and distribution, determine the routes and rates of excretion, the mass balance recovery of the 14C material from the urine, the feces, and also expired air as applicable. And then also to assess the metabolite profile and metabolite identification. So usually 6 subjects would be recruited to the study. However, it may be necessary to increase the numbers of subjects if your IMP is known to alter GI transit or motility, or if the non-clinical data were highly variable. In addition, fewer subjects may be needed if you are conducting the study in oncology patients, in which case the data are analysed on a case-by-case basis, so you can determine whether or not each subject is eligible and whether whether the subject is evaluable, and therefore you need to ensure that you have 3 to 4 evaluable profiles to be able to determine the objectives of the study. The majority of studies are conducted in healthy male subjects, and they are typically aged between 35 and 65, although there are cases where it may be possible or necessary to include female subjects. Subjects should be excluded if they are regularly or recently exposed to radiation. So although the levels of radiation that would be administered in a human AME study are relatively low, the potential for a higher cumulative dose in subjects who are already exposed to radiation should be avoided. In addition, subjects who should have regular bowel movements, and during the study they'll receive a high fibre diet that's low in indigestible foods. So the study itself usually involves a screening period, typically 28 days, followed by checking in on day -1 and dosing on day 1. And the subjects would then remain in-house whilst all the blood, urine and fecal samples are collected. The duration of the in-house stay may vary depending on the expected rate of elimination and the observed rate of elimination, with a minimum and maximum duration defined within the protocol. So we'll come on to that in a little bit more detail around how we determine the minimum and maximum confinement period. So typically this should be around or at least 5.5 times the longest half-life. So the half-life of either the parent or any known metabolites. So that's to ensure that the profile can be fully defined. Once the minimum stay has been reached, then the discharge criteria defined within the protocol can be used to determine whether a subject should remain confined for longer, and therefore the collection of the urine and faeces for a longer period. So typical discharge criteria that you may see in a protocol are greater than 90% of the total dose recovered, less than 1% in 2 consecutive 24-hour periods is recovered in the urine and the faeces. And in addition, you want to check that the plasma radiation has fallen below the limit of detection for at least 2 consecutive collections. So the samples for radioanalysis are shipped daily to Metabolism, where they are analysed immediately so that we can get the results back to the site within 24 hours to determine whether or not the subjects have met the discharge criteria and therefore can be released from the site. Once the maximum confinement period is met, then the subjects have to be discharged. So for compounds where you anticipate that there is a longer half-life, then it may be possible to, initially to extend the in-house stay, but also to include additional weekly or bi-weekly, tri-weekly in-house visits to collect additional 24-hour collections, rather than have the subjects remain in-house for the entire duration. So this should not occur until it is believed that the linear elimination phase has been reached, and therefore you can extrapolate between the collections performed. However, if precise mass balance is desired, it would still be preferable to keep the subjects confined continuously, and we have had subjects confined for up to 144 days to ensure that we can obtain mass balance. And where recovery is low at the later time points, it may be possible to switch to AMS analysis to follow the elimination. So the figure on the screen shows an example of a plasma concentration profile where the half-life of the total radioactivity is significantly longer than that of the parent due to slow forming and persistent metabolites. So this highlights the importance of having the plasma discharge criteria in addition to the recovered dose discharge criteria, so that we ensure that we're following out the radioactivity until it falls below the limit of detection. So critical to the success of a human AME study is having an on-site GMP pharmacy who are able to handle radiolabeled material. So depending on the preference of the sponsor and the stability of the product, that can involve either the manufacture of the drug product at site or the receipt of drug product for assembly into unit doses. And this includes the manufacture of non-sterile solid and liquid dose forms for oral or topical use, and also the manufacture of sterile solutions for injection or infusion. So, as discussed with John earlier, the radioactive dose administered should be as low as reasonably achievable to meet the objectives of the study. And in general, the dose that we administer would be in the category 2A, so 0.1 to 1 millisievert. However, it is possible to justify going slightly above that range and up to 2 millisieverts, so into the category 2B criteria. And so you can see here that in the last 5 years, 89% of the studies that we've conducted have met the 2A criteria, so being less than 1 millisievert. However, 11% have been between 1 and 2 millisieverts. And in fact, the highest radioactive dose that we've been able to justify to the RSAT committee to date has been 1.7 millisieverts. Also presented here are some typical recovery data from some recent studies. In general, the recovery is greater than 90%, with varying contributions from urine and feces, with a number of compounds being almost exclusively eliminated fecally. And just to highlight one study where recovery was low, so there's a study where the total recovery was 82.5%. And for this study, actually, none of the subjects had met the discharge criteria on the maximum confinement period of day 15 due to an unidentified persistent metabolite. Fortunately, this study protocol did allow for up to 2 additional visits to occur after the discharge criteria to allow for collection of samples for another 24-hour period. So all the subjects returned on day 21 and day 28 for additional recovery data to be assessed. However, it was noted that all still had radiation at day 28, and so the overall recovery was low. However, we were able to perform extrapolation from the data from those additional outpatient visits. And then finally, I just want to come on to sample collection. So accurate sample collection is again critical to the success of an AME study. You know, this is what gives you all of the information at the end of it. And so making sure that you plan correctly to collect the data at the right time points and handle the samples correctly throughout the study is critical. So urine samples are generally collected over smaller collection periods at first, and then 24-hour collections once we get out of the initial elimination phase. And fecal samples are also collected over 24-hour periods, and both the urine and the feces will continue collection until the discharge criteria are met. Blood samples will be collected for radioanalysis in both the plasma and in whole blood, so that we can look at the association of the radioactivity to blood cells. And the sample times for these samples would follow a typical PK profile, and again would continue until the discharge criteria are met. Expired air is generally not required. However, it may be required if the 14C label is in a liable position on the molecule, or the IMP or drug class are known to have an excretion by expired air. If expired air samples are required, then they're collected using a trapping agent, and typically the timings would follow the PK profile out to around 48 hours post-dose. If a subject vomits within 48 hours of dosing, then ideally we would collect all of the emesis and determine what the recovery is, so that we can determine whether or not the subject's data is likely to be evaluable. In addition, plasma samples for standard bioanalysis would be collected over the normal PK profile for the parent and any known metabolites. Samples for metabolite profiling would also be collected using a refined PK sample profiling, but ensuring that samples continue to be collected throughout the entire residential period to cover any late emerging or persistent metabolites. Due to the numbers of samples that need to be collected, then blood volume is always a consideration that needs to be taken into account fairly early in the development of a human AME protocol. So ideally, the total blood volume should not exceed 500 mils per subject. So now I'll pass back to John, who's going to talk a little bit more about protocol metabolite identification.
Thanks, Kath. So the major final deliverable at the end of the human AME trial is, of course, to identify and quantify the metabolites present in the plasma, the urine, and the feces samples collected to address that regulatory guidance narrative that we highlighted earlier, at the start of this session. Now, this often takes place during and following database lock for the primary endpoints of a clinical phase, so after the radioanalysis and the PK determinations have been completed. And the phase report is often appended to the final clinical report when the metabolite investigations are complete. So an important factor to the success of this metabolite identification is building upon the experiences with the non-clinical samples generated during that rat or non-rodent ADME study. And the benefit of the same lab, the same teams, overseeing the extraction, the concentration, the analysis of those clinical samples, and of course the interrogation and interpretation of the data as those that have conducted the non-clinical analyses should not be overlooked at all. So this simple graphic illustrates the process of metabolite profiling and ID that I'm sure we're all familiar with. And with the advances in the sensitivity and the selectivity of high resolution mass spectrometers over the past 10 years or so, the expectation is that we do identify all of those metabolites that may contribute to the overall exposure in humans. In fact, we typically go down well below the 10% level indicated in the regulatory guidance and go as low as 1% of those metabolites circulating systemically, and similarly around 1% of those present in the excreta. We're also still asked to present data as percent total radioactivity and percent parent contribution, which of course was a legacy of earlier guidance requests by both the FDA and the EMA. However, with those metabolite investigations being relatively resource intensive, a number of strategies have been employed to reduce the number of samples analysed while maintaining maximal data available and obtained from those subjects. Not all samples collected from a subject will be individually analysed, for instance. And if PK and excretion profiles from all individuals in a study are similar, plasma may be pooled across subjects and across time points at Cmax, Cmax plus T half, for instance. And a plasma AUC pool can also be prepared to generate a single sample per subject that's representative of the relative total exposure of the parent and metabolites over the course of the study in a single sample. Equally, urine and feces may be pooled proportionally across subjects at each time point, the 0 to 24, 24 to 48 hours, for instance. And this again reduces the number of urine and feces pools submitted for further analysis. But ultimately, the quantification and the identification of the metabolites in humans better informs us as drug development scientists in decision making and helps de-risk projects by identification of those potentially disproportionate or unique metabolites before those large-scale patient studies are conducted. So we touched upon the challenge of setting an appropriate radioactive dose earlier in order to meet the scientific objectives of the study, while considering obviously the risks of the radiation burden to the subjects. And this remains a primary challenge to metabolite identification scientists too, obtaining those samples with sufficient radiolabeled material within them above detectable limits to quantify and characterize constituents completely. So in order to assess the dose and justify any necessary increase to RSAC, we follow a defined strategy in the lead up to any human trial. The dosimetry calculated value is utilized with other available data to illustrate the likely concentration of circulating metabolites and the limits of detection from samples that we expect to obtain in a human trial. For this, we'll use the proposed clinical dose levels, the cold dose as well as that of the radiolabeled material. We'll use the specific activity of the clinical dose formulation, We'll use any previous human PK data from non-labeled SAD or MAD studies. We'll look at the expected level of metabolism based on both previous in vitro and preclinical in vivo ADME studies. And we'll use sample preparation and concentration techniques and the knowledge obtained in earlier studies working with the compound to think about how we extract and prepare samples for analysis. And since the dosimetry calculation provides a linear scale for the amount of radioactivity in terms of microcuries, which would provide a body burden in terms of millisieverts, we could work through what radioactive dose level, what specific activity, and what total amount of radioactivity would provide a total body burden in that region of 1 to 2 millisieverts. As you recall, the ICRP 2A or 2B category, and whether an increased radioactive dose would actually lead to a better sensitivity of detection of those radiolabeled metabolites throughout the study period. If, however, considering all of these strategies and increasing the carbon-14 material poses too great a risk to the subjects, and alternative analytical endpoints such as that of AMS, accelerated mass spectrometry, can be used for both the radioanalysis and the metabolite profiling, where those radioactivity levels are too low for conventional radio detection. So hopefully today we've gone through many of the elements that make up a human AME study, but it may also be helpful just to put that into context of some of the challenges that are faced in a real life example. So in this scenario, we faced that during a clinical development of a molecule, metabolite identification investigations identified potentially disproportionate human metabolites. And these were highlighted as acyl glucuronides. Now, unfortunately, this was significantly different than the metabolite profile from the preclinical toxicology species obtained from both in vitro and in vivo investigations. Now, as we touched on earlier, the misguidance highlights this is of concern, and the clinical studies were put on hold while the metabolite strategy regarding quantification was considered. Now, Covance confirmed the presence and structure of these putative metabolites as indeed acyl glucuronides, but without analytical standards at the time, obviously we couldn't quantify them yet in the clinical samples. So to what level they circulated at, in response to the regulatory guidance was unknown. So between the client, Covance, and Arcinova, of course, the synthesis and planning of the carbon-14 labeled material, the non-clinical study conduct, the dosimetry, and the regulatory clinical trial application submission were coordinated with parallel activities to ensure shortest timelines as possible. Data review of the existing parent data that was available, assumptions on the putative metabolite levels, and dose calculations of the drug and the 14C to be administered helped define not only a pooling and analytical strategy, but also justification for increased carbon-14 dose level for RSAC approval and an increased blood volumes at some critical time points to fully allow for metabolite profiling and ID to move forward. Now, the schedules and dedicated resources were locked down with an aggressive timeline in mind, and weekly project calls established a rhythm of progress updates, which allowed schedules to be accelerated further as data was generated and reviewed rapidly. So the use of the radio-labeled material enabled full quantitation of the acyl glucuronides of the parent molecule, as well as other circulating metabolites, and these were demonstrated to actually be below the 10% threshold trigger for further non-clinical toxicity testing of those metabolites. So the successful outcomes were based upon, I suppose, an iterative knowledge acquisition from the initial high-resolution mass spectrometry data to help align the radioisotope position selection, as well as then the knowledge from the conduct of the non-clinical studies for sample handling and profiling, allowed us to plan for what-if scenarios and actually consider backfilling data should the 10% threshold be exceeded and alternative non-clinical safety testing in alternative species was required. And of course, in this example, the clinical studies were then continued. So in conclusion, regulatory bodies are continuing to demand a greater extent of metabolism information earlier to help de-risk any safety concerns and reduce compound attrition in later stage trials. Now, planning your human study early while considering preclinical ADME and radiosynthesis components will help enable the clinical study design and conduct be considered more roundly at an earlier stage. And the largest single factor contributing to meeting any aggressive timeline is the availability of the quantity of GMP radio-labeled drug and, of course, its stability. So working backwards with all of these components in mind helps in planning. Experienced scientists, clinicians, and project teams are all needed to overcome unique challenges encountered on these human AME studies, and using this experience helps foresee and mitigate issues. And finally, a single outsourcing strategy can enhance the speed of delivery and understanding of those components from the radiosynthesis, the preclinical work, pharmacy elements, and of course, the clinical conduct and analyses. So thank you very much for your attention today. I'll now hand back to John for some closing remarks and to coordinate any questions.
Okay. Well, thank you, John. Thank you, Kath. Thank you, Ellie, for very detailed, informative presentations. So just a reminder to everyone, we will send out the presentation after this call. At the end of the presentation will be contact details for Kath, Ellie and John, should you have any questions you would like to follow up individually with them. And also a reminder, if you have a question for this session, please use the chat function. I'll be happy to read out the question to John, Ellie and Kath. So a couple of questions. So if I can just, sorry, excuse me while I scroll up to the first one. Probably for you, Ellie, this one. So studies performed with doses of less than 1 microcurie. Should radio-labeled IP synthesis also be to GMP?
Great. Yeah, no, thanks for that, John. It's a really good question. And when we're considering microdoses, where we're obviously administering a very low amount of radioactivity, we'd still recommend that the synthesis is performed to GMP because you're obviously administering the material to healthy volunteers. But there is obviously less risk associated with this than if you're giving a macro dose. But our preference is still to do it to GMP. It also comes down to the discretion of the QP at the clinical unit. And most of the European clinical units have a GMP grade pharmacy, so they won't often accept non-GMP material into them as well for dosing. So it's a discussion to be had with both the radiosynthesis provider and the clinical unit.
Okay, thanks, Ellie. So the next question, in non-clinical mass balance studies, rodents, non-rodents, which dose level do you select? The no overall adverse effect level or a dose below that?
I could start on that one, but I think Kath also would be able to share some thoughts, John. So I think we've tried to describe today a number of considerations in setting both a radioactive dose level in particular, but if you look at it in context, the actual dose of that API also has an impact on your scientific endpoints. So it has the impact on the specific activity, as I think Ellie alluded to. So in general, most of the clinical studies that we conduct with radio-labeled materials do actually go in at I suppose, a therapeutic type dose. So that kind of fits into the therapeutic window of, the SAD and MAD studies and the other clinical studies that are conducted, so that data can be directly extrapolated and compared. However, you still need to justify the selection of that dose level, that you're going to have a sufficient limit of detection of both, parent and metabolites. So it's the radioactivity and the specific activity that also go into that consideration. Kath, do you want to add a little bit more to that as a response?
I think that covered it really, John. So I think, yeah, as you say, from a human AME perspective, we would be looking to use a therapeutically relevant dose level, but also taking into consideration, as John mentioned, really what the specific activity is going to be, and ensuring that the radioactivity will be sufficient to meet those endpoints. So it's a collaborative team effort, I would say, discussions with both Arcinova and the metabolism group and the clinic itself to determine what would be a suitable dose for the human AME trial.
Okay, thank you. The next question, when should we consider multiple dosing in a QWBA study?
Well, that's a good question. If you look back at the missed guidance, and part of what I didn't describe, I suppose, was that systemic exposure in terms of metabolites for safety consideration are usually done in the context of steady state kinetics. So we do occasionally get asked about, should we do repeat dose ADME studies per se, and especially, then should you actually even look at steady state kinetics for a repeat dose human study? Obviously, there are implications for such an approach in terms of, the amount of radioactivity that could be administered to humans, but certainly in animals, there are no restrictions in terms of the amount of radioactivity that you could deliver either as a single dose or as repeated doses over, a relatively short period of time, such as a week. So it's theoretically possible to do. However, we don't typically follow that approach. There's no regulatory consideration to say you have to do a tissue distribution and therefore look at accumulation of radio-labeled metabolites or parent for that dosimetry calculation, and a single dose is the typical approach for generating data for that single dose study then in humans. So I think if there was a requirement or a request to do a repeat dose study in humans, then again you would probably match that with a repeat dose study in your rodent tissue distribution and excretion balance. But in general, the human doses are single dose, and therefore you mirror that in your animal study as well.
Okay. And I think touching on that, so how to proceed with a human mass balance study when an active metabolite cannot be fully assessed following a single dose?
Oh, I think I need a little bit more context to address that one, and maybe that's one we could follow up offline at a later date. I suppose it means, I'd like to explore what it means in terms of can't be fully assessed. Is it limited detection? Is it, what is that? What does that mean? So I'm sorry, unless we can, I know we're not going to open up chat for general consideration, but yeah, maybe my contact details at the end, and let's have a follow-up discussion on that one.
Yeah, sure. Okay. So that was the last question that we had. So I guess just to close, again, John, Ellie, Kath, thank you very much for your presentations and answering the questions as well. Thank you, everyone, for taking the time to dial into this session. Both Covance and Arcinova hope that you found it informative, and we'd be very happy to follow up with any subsequent questions that you have. So with that, I'll close the call, and thank you very much, everyone, for your attention.
Thank you all.
Thanks, everyone.